NCERT based single sentence questions, molecular basis of inheritance. Let's start. Long polymer of deoxyribonucleotides is DNA. Length of DNA usually defined as number of nucleotides. Number of nucleotides. Number of nucleotides in pi into 174. 5386. Number of nucleotides in lambda bacteriophage. 48,502. Number of nucleotides in E. coli. 4.6 into 10 to the power of 6. 10 to the power of 6 base pairs. 10 to the power of 6 base pairs. Pair of nucleotides usually referred to as base pairs. The three components of nucleotides are nitrogenous bases, pentose sugar, and phosphate group. Pentose sugar of RNA, ribose sugar. Pentose sugar of DNA, deoxyribose. Number of nucleotides in haploid content of human DNA, 3.3 into 10 to the power of 9 base pairs. 10 to the power of 9 base pairs. Nitrogenous base is linked to the pentose sugar with the help of N-glycosidic linkage. N-glycosidic linkage. When a phosphate group is linked to 5-OH of nucleoside, it forms nucleotide. 5 end of polynucleotide chain means free phosphate end of ribose sugar. Free phosphate end of ribose sugar. The two types of nitrogenous bases are purines and pyrimidines. Purines and pyrimidines. Adenosine or deoxyadenosine means adenosine or adenine plus pentose sugar. Nitrogenous base plus pentose sugar is considered as nucleoside. Purine nitrogenous bases are adenosine. Purine nitrogenous bases are adenine and guanine. Nucleoside plus phosphate group is considered as nucleotide. Pyrimidine nitrogenous bases are cytosine, uracil, and thymine. Cytidine or deoxycytidine include cytosine plus pentose sugar. Deoxynucleotide means nucleoside plus phosphate. Pyrimidine, which is seen only in DNA, that is thymine. Pyrimidine, which only seen in RNA, that is uracil. Guanosine or deoxyguanosine is guanine plus pentose sugar. Pyrimidine, which is common for both DNA and RNA, cytosine. N-glycosidic linkage links nitrogenous base to pentose sugar. Link nitrogenous base to pentose sugar. Phosphodiester linkage links phosphate group to nucleoside. In RNA, thymine is replaced by uracil. Uridine belongs to RNA. Nitrogenous base of DNA in the place of uracil of RNA, thymine. Deoxythymidine include thymine plus pentose sugar. Phosphodiester linkage links two nucleotides. Three end of polynucleotide chain means free 3 OH group. Free 3 OH group at the end of ribose sugar. Nucleoside include pentose sugar plus nitrogenous base. The backbone in a polynucleotide chain is formed by sugar and phosphate. Polynucleotide include many nucleotides. Nucleotide include nucleoside plus phosphate. Nucleoside include nitrogenous base plus sugar. Two nucleotides are linked through 3,5 phosphodiester linkage. Phosphate group linked to nucleoside through phosphoester linkage. Nitrogenous base linked to pentose sugar through N-glycosidic linkage. N-glycosidic linkage. Scientists who provided X-ray diffraction data on genetic material are Morris Wilkins and Rosalind Franklin. Morris Wilkins and Rosalind Franklin. Scientists who proposed the double helix model of the structure of DNA are James Watson and Francis Crick. Scientists who proposed that the ratios between adenine and thymine are constant and equals 1. Chargaff. Scientists who proposed that the ratio between guanine and cytosine are constant and equals 1. Chargaff. Year which double helix model proposed? 1953. Year which DNA identified as acidic substance? 1859. Basis for the double helix model of DNA is X-ray diffraction data. X-ray diffraction data and Chargaff rule. DNA in double helix model made with two polynucleotide chains. If one chain has a polarity of 5' to 3', the other has 3 to 5'. Such phenomena is called anti-parallel polarity. Bond between base pairs, hydrogen bond. A nanometer in the meter is 10 to the power of minus 9. 10 to the power of minus 9. Distance between the two consecutive base pairs of DNA is in nanometer, 0.34 nm. Distance between the two consecutive base pairs of DNA in meters, 0.34 into 10 to the power of minus 9 meters. 10 to the power of minus 9 meters. Always purine comes opposite to a pyrimidine. This generates uniform distance between the two strands of helix approximately. The two chains of DNA are coiled in right-handed fashion or left-handed? Right-handed fashion. Number of base pairs in each turn. Number of base pairs in each turn. 10 base pairs. Pitch of the helix is 3.4 nanometer. Approximate distance between the base pairs in helix 0.34 nm. Genetic information flows from according to the central dogma. Genetic information flows from according to the central dogma, that is DNA, from DNA to RNA to protein. Chemical nature of DNA was discovered by Frederick Miescher. Chemical nature of DNA is acidic. Number of hydrogen bonds between adenine and thymine 2. If one DNA strand with purine, the opposite complementary strand with pyrimidine. Length of DNA double helix in a typical mammalian cell 2.2 meters. Number of hydrogen bonds between guanine and cytosine 3. Scientists who named the DNA as nuclein, Frederick Miescher. Central dogma in molecular biology is proposed by Francis Crick. In some viruses, the flow of information is in reverse direction, that is from RNA to DNA. Can you suggest a simple name to the process? Reverse transcription. Dimension of nucleus in a typical mammalian cell. Dimension of nucleus in a typical mammalian cell is 10 to the power of minus 6, 10 to the power of minus 6 meters. Length of DNA in Escherichia coli 1.36 meters. The dash equal to total number of base pairs into distance between the two consecutive maps. Total length of double helix. Total length of double helix DNA in organism. Dash equal to distance between the two consecutive maps by total length of double helix of DNA in an organism. Total number of base pairs in any organism. Electrical charge of histone protein is positive. Charge of protein depends on abundance of amino acid residues, with charged side chains. Amino acid rich in histone, lysine and arginine. Name given by Frederick Miescher for DNA is nuclein. Process of formation of DNA from the DNA is called replication. Process of formation of mRNA from DNA called transcription. Process of formation of protein from mRNA is called translation. Electrical charge in DNA negative. Electrical charge in proteins around DNA positive. Arrangement of DNA in nuclei organized in large loops held by proteins. Electrical charge which lysine and arginine carry in their side chains, positive. Histone are rich in the basic amino acid residue. The basic amino acids are arginine and lysine. The negatively charged DNA is wrapped around the positively charged histone aptamer to form a structure called nucleosome. Thread-like strained bodies of nucleus are called chromatin. Region of chromatin which are loosely packed and less strained is considered as euchromatin. The chromatin that is more densely packed and stained dark is called heterochromatin. Transcriptionally active chromatin, euchromatin. Transcriptionally inactive chromatin, heterochromatin. Number of base pairs in the typical nucleosome of the DNA helix, 200 base pairs. Nucleosomes constitute the repeating unit of structure in nucleus called chromatin. By this year, the quest to determine the mechanism for genetic inheritance had reached to the molecular level, 1926. When viewed under electron microscope, the nucleosomes in chromatin are seen as beads on string. Dash and numerous other scientists have narrowed the search to the chromosomes located in the nucleus of most cells. Thomas Hunt Morgan. Beads on string structure in chromatin are packaged and form this structure. Chromatin fiber. Transforming principle proposed by Frederick Griffith. Nitrogenous bases which are considered as pyrimidines, cytosine and uracil. Nitrogenous bases which are considered as pyrimidine. Nitrogenous bases which are considered as purines, adenine, guanine. Pyrimidines, cytosine and uracil. Pyrimidine which is exclusively belongs to DNA, that is thymine. Here we have to write thymine also. Pyrimidine which is exclusively belongs to RNA, uracil. Nitrogenous base plus pentose sugar is called nucleoside. Nucleoside plus phosphate is called nucleotide. Nucleotide plus nucleotide is called dinucleotide. When more nucleotides join each other, called polynucleotide. Complete series of polynucleotide chain of an organism which is arranged in the form of thread-like fiber is called chromatin. Condensed chromatin during metaphase of cell division is called chromosome. The beads and string structure in chromatin packaged to form chromatin fiber that are further coiled and condensed at metaphase stage are called chromosomes. Condensed chromatin is called chromosome. Chromatin fibers are packaged beads. Chromatin is repeated nucleosome. Nucleosome is DNA part which wrapped around histone aptamer. Base pairs in typical nucleosome, 200 base pairs. The package of chromatin at higher level require additional set of protein that collectively are referred to as non-histone chromosomal proteins. Non-histone chromosomal protein. Experimental organisms of Griffith: Streptococcus pneumoniae. Streptococcus pneumoniae. Principle explained by Griffith. Transforming principle. Scientists worked to determine the biochemical nature of transforming principle in Griffith experiment. Oswald Avery, Mac Leod, and Macklin McCarthy. The unequivocal proof that DNA is the genetic material came from the experiments of these scientists: Alfred Hershey and Martha Chase, 1953. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. That name the scientists who said this: Watson and Crick. Bacteria used in transforming principle. Bacteria used in transforming principle is Streptococcus pneumoniae. The two strains growth on culture plate for Streptococcus pneumoniae are rough strain and smooth strain. Rough strain and smooth strain. Virulent strain of Streptococcus is smooth strain. Non-virulent strain of Streptococcus is rough strain. Reason for the virulent nature of smooth strain is they have polysaccharide mucus coat. Reason for the non-virulent nature of rough strain: they lack polysaccharide mucus coat. A strain injected to mice dies. R strain injected to mice live. A strain, mice survive. Let us say it as mice survive. A strain, heat killed injected to mice survive. A strain, heat killed plus R strain live injected to mice dies. Heat killed a strain plus R strain live injected to mice, recovered bacteria and dead mice are with a strain. When heat killed a strain along with R strain injected into mice, how live strains are recovered in dead mice? That we say R strain bacteria somehow being transformed by the heat killed strain. In transforming principle, what is the conclusion given by Griffith? Griffith conclude genetic material is transformed. Genetic material is transformed from heat killed strain to R strain. Biochemical characterization of transforming principle was proposed by proposed by Oswald Avery, Colin MacLeod, Macklin McCarthy. Macklin McCarthy. Scientists continued the experiments of Griffith transforming principle. Scientists continued the experiments of Griffith transforming principle. The same group of scientists, we can say. Prior to the work of these scientists, the genetic material was thought to be a protein. Oswald Avery, Colin MacLeod, and Macklin McCarthy. Biochemicals used to treat the heat-killed strain by Oswald Avery, Colin MacLeod, Macklin McCarthy are DNAs, RNAs, proteases. Heat-killed strain plus are treated with protease injected to mice along with R strain, mice die. Heat kill strain plus treated with RNAs injected to mice along with R strain, mice die. Heat kill strain plus treated with amylase injected to mice along with R strain, mice die. Heat kill strain plus treated with DNAs injected to mice along with R strain, mice survive. Major components of heat kill strain are carbohydrates, proteins, DNA, and RNA. When enzymes like amylase, protease, RNAs are applied, it removes these components of heat kill strains respectively: carbohydrates, proteins, and fat, RNA. If heat kill strain is treated with DNase enzyme, it removes these components of heat kill strain, DNA. Heat kill strain treated with protease, amylase, RNAs and injected to mice along with R strain, and the mice die. What is the conclusion of Avery, MacLeod, McCarthy from the above experiment? Transformed material is from heat killed strain to R strain is DNA. Avery, MacLeod, McCarthy discovered that protein digesting enzymes and RNA digesting enzymes did not affect transformation. So the transforming substance was not protein or RNA. Enzyme which inhibit transformation is DNAs. What is the difference between the DNA and DNAs? DNA and DNAs. DNA is substrate and DNAs is enzyme. Experimental organism of Alfred Hershey and Martha Chase, bacteriophages. Viruses that infect bacteria are called bacteriophages. Hershey and Chase worked to discover whether it was dash a dash from the viruses that entered the bacteria, protein or DNA, according to them. Hershey and Chase grew some viruses on a medium that contained radioactive dash and some others on medium that contained radioactive dash, radioactive phosphorus and radioactive sulfur. Inorganic element of DNA. Inorganic element of DNA, phosphorus. Inorganic element of protein, sulfur. Viruses grown on radioactive sulfur contain and do not contain. Viruses grown on radioactive sulfur contain radioactive sulfur and do not contain radioactive DNA. Viruses grown on radioactive phosphorus contain and do not contain radioactive DNA and do not contain radioactive protein. Radioactive protein and radioactive DNA. Bacteria which was infected with virus were grown in the radioactive phosphorus medium or without radioactive DNA. Bacteria which was infected with virus were grown in the radioactive sulfur medium or without with radioactive DNA. Without radioactive DNA. Bacteria which was infected with virus that has radioactive DNA were radioactive. It indicates DNA was the material that passed from virus. Bacteria that were infected with viruses that had radioactive protein were not radioactive. It indicates proteins did not enter the bacteria from the viruses. Genetic material in tobacco mosaic virus is RNA. Genetic material in QB bacteriophage is RNA. A molecule that can act as a genetic material must fulfill the following criteria. Fill in the blanks with reference with respect to the given statement. It should be able to generate its replica. It should dash and dash be stable chemically and structurally. It should be able to express itself in the form of Mendelian characters. It should be able to express itself in the form of Mendelian characters. It should provide the scope for, here it is, wait a minute, Mendelian characters. It should provide scope for slow changes, here it is, that is nothing but mutations. Stability is one of the properties of genetic material. Which principle is strongly support this statement? Transformation principle of Griffith. Heat killed a strain but cannot damage the DNA. What happened when heated? DNA strands are separated. Heat killed the strain but cannot damage the DNA. What happened when appropriate conditions are provided? Appropriate conditions are provided, united with their complementary strands. With their complementary strands. Heat killed the strain but cannot damage the DNA. Which property of genetic material it shows? Stability. Reactive group of RNA is 2OH at every nucleotide. It makes RNA labile and easily degradable. Highly reactive group 2OH, highly reactive group of 2OH on every nucleotide. RNA is known to catalytic, hence reactive. Better genetic material among the two nucleic acid, DNA. Additional stability of DNA due to this property with respect to nitrogenous bases, that presence of thymine. Mutation rate faster in RNA or DNA? RNA. Viruses have shorter lifespan. Mutate, evolve faster. Why? Why? They have RNA as genetic material. Why RNA easily express the characters than DNA? RNA can directly code for the synthesis of RNA, then directly code for the synthesis of proteins. Mutation rate is faster in RNA when compared with DNA. Why? RNA is unstable. Protein synthesizing machinery has evolved around this genetic material. Protein synthetic machinery has evolved with this genetic material, RNA. Preferred genetic material for the storage of genetic information. Preferred genetic material for the storage of genetic information, that is DNA. Preferred genetic material for the transmission of genetic information is RNA. First genetic material is RNA. There is now enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc. evolved around RNA. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. The scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. Semi-conservative semi-conservative scheme of replication. Heavy isotope of nitrogen, N15. Light isotope of nitrogen, N14. Time taken for Escherichia coli for each multiplication, 20 minutes. Genetic material which acts as catalyst also, RNA. Double strand nature, having complementary strand further resists changes by evolving a process of repair, made this genetic material more stable, DNA. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative replication, semi-conservative method of method of DNA replication. RNA as a catalyst unstable because it is highly reactive. Name the scientist, it has not escaped our notice that the specific pairing having have postulated immediately suggest a possible copying mechanism for the genetic material. Watson and Crick. Watson and Crick in 1953. Name the scientist semi-conservative method experimental proof first time in Escherichia coli. Matthew Meiselson and Franklin Stahl. To incorporate N15 in the DNA of Escherichia coli, this culture has to be done. They grew Escherichia coli in a medium containing NH4Cl, N15 radioactive isotope with NH4Cl as the only source for many generations. Two strands of DNA with N15 are grown and NH4Cl N14 medium result after 20 minutes. After 20 minutes, DNA found with hybrid or with intermediate density. That in that one strand is with N15 and other strand, one strand with N15 and one strand with N14. Equal amounts of hybrid DNA and light DNA are found after 40 minutes. Second generation. N15 containing DNA grown in N14 medium after 20 minutes replicated. Write the condition of each strand. Total of 2 DNA replicated. First DNA strand. First DNA strand 1 is with N15. In the first DNA, strand 2 is with N14. Next, second DNA strand. Strand 1 is with N15. Strand 2 is with N14. Strand 2 with N14. In Michelson and Stahl experiment, condition of DNA strands in E. coli after 40 minutes. Total 4. Double helical strands, total 4 double helical strands. In that, total 4 double helical strands. In that, first DNA strand with N15, second with N14, second DNA strand, both strands are with N14, third DNA strand, both strands are with N14, fourth DNA strand, One strand with N15, one strand with N14. Experimental proof, semi-conservative method on Vicia faba is done by Taylor and his colleagues. Taylor and colleagues in 1956. Average rate of replication or polymerization in E. coli approximately per second 2000 base pairs per second. Time taken for the completion of process of replication of total content of DNA 4.6 into 10 to the power of 6, that is 18 minutes. Energetically replication is very expensive process. In addition to acting as substrate, they provide energy for replication. Deoxyribonucleoside triphosphate used as dual purpose in replication process. Deoxyribonucleoside triphosphate. The two terminal phosphates in a dash are high energy phosphate, same as in case of ATP, that is deoxyribonucleoside triphosphate. DNA dependent DNA polymerase catalyze polymerization only in one direction, that is in 5' to 3' direction. 5' to 3' direction. Additional complications at the replication fork are created due to this enzyme and its property. What is the enzyme? What is the enzyme? Dependent DNA polymerase. What is the property? Property, it catalyze the polymerization. It catalyze polymerization in only one direction, that is 5'3" direction. That is 5'3" direction. What are the complications? On one strand, replication is continuous and discontinuous on the other strand. Discontinuous on the other strand. If in replication fork, the template with polarity 3,5, the replication is continuous. In replication fork, the template with polarity 5,3, the replication is discontinuous. Replicated strand of DNA which grows continuously without any gap is called leading strand. Replicated strand of DNA which is formed grows discontinuously with gap is called lagging strand. Replicated strand of DNA which need a separate RNA primer to start, lagging strand. Replication strand of DNA which need a single RNA primer to start its growth, leading strand. Replication strand of DNA which does not require DNA ligase enzyme, leading strand. Replication strand which require DNA ligase enzyme, lagging strand. Replication strand of DNA in which direction of growth is 5'3", leading strand. Replication strand of DNA which direction growth is 3'5", lagging strand. Replication strand of DNA where Okazaki fragments are formed. Lagging strand. Can replication process can be initiated randomly at any place of DNA? No. Is there any specific reasons are present on DNA for the initiation of replication? Yes. Place of origin of DNA replication in E. coli are called as origin of replication, ori site. During cloning of vector, ori site is important. Why? Because any sequence linked to ori, because any sequence linked to ori in a plasmid vector will get replicated. The sequence helps in initiation of replication. A foreign piece of DNA has to be linked to the sequence in order to replicate it within the host cell. It also controls the copy number of linked DNA of ori site. That is, ori site or origin of replication. Origin of replication in prokaryotes. The site of replication in prokaryotes. The site of replication in prokaryote, that is in cytoplasm. The site of replication in eukaryote, nucleus. Phase of replication in eukaryote during cell division, S phase. Replication of DNA and cell division cycle should be highly coordinated in eukaryotes. A failure of cell division after DNA replication leads to a chromosomal aneuploidy, a chromosomal anomaly called polyploidy. DNA replication is a multi-step complex process which require DNA replication, a multi-step complex process which require over a dozen of enzymes, protein factors, and viral DNA. Single oocyte is present in eukaryotes. Multiple oocytes are present in eukaryotes. Single oocyte is present in prokaryotes. Reason for multiple oocytes. Reason for multiple oocytes due to larger size and association with proteins. Whole DNA act as a single replicating unit in prokaryotes. Several replicating units or replicants are seen in eukaryotes. Reason for the whole DNA acts as a single replication unit due to single oocyte. Reason for the presence of multiple replications or several replicating units due to the presence of several oocytes on DNA. Site of origin of replication and site of termination of replication are respectively called oocyte and terminus. Site of terminus is site of origin of replication in site of terminus is site of origin of replication in prokaryotes. Site of origin of replication and site of termination of replication are same in circular DNA of prokaryotes. DNA replication can be unidirectional or multidirectional, bidirectional, both. Direction of replication, unidirectional or bidirectional, depends on whether the replication from the point of origin proceeds in only one direction or both directions. Initiation of replication takes place due to multi-enzyme complex called replisome or replication apparatus. Deoxyribonucleoside monophosphate, which serves as a raw material in DNA synthesis for adenine, DAMP, that is deoxyriboadenosine monophosphate. Deoxyribonucleoside monophosphate, which serves as a raw material in DNA synthesis, DTMP, deoxyribothiamidine monophosphate. For guanine, DGMP, deoxyriboguanosine monophosphate. For cytosine, DCMP, deoxyribocytidine monophosphate. All the deoxyribonucleoside monophosphates are found floating free in nucleoplasm and serve as the raw material for DNA synthesis. Replisome or replication apparatus is multi-enzymes complex for initiation of replication. In the presence of energy, the ATP and can enzyme phosphorylase the nucleoside DAMP, DGMP, DCMP, TMP, phosphorylated and changed into triphosphate states respectively like this: DATP and DATP, DGTP, DCTP, DTTP. The DNA molecules is intricately coiled into a chromosome and its unwinding is not an easy process. The replication starts with an incision or cut, generally referred to a nick at specific point, one of the two parental strands. The nick is brought about by incision enzyme called topoisomerase. Topoisomerase. During DNA replication, whole of DNA does not open in one stretch, but the point of separation process slowly from one end to the another. It gives the appearance of Y-shaped structure, hence called replication fork. Replication initiates at the 3 end to 5 end. 3 end or 5 end, that is 3 end. Initiation of new DNA synthesis requires a short piece 10 to 15 nucleotides long of RNA and it is called RNA primer. For the initiation of new DNA synthesis, dash is synthesized at the dash end of each template with the help of special RNA polymerase. RNA primer at 3N and the help of a special RNA polymerase called primer. In replication fork, RNA primer lies at the free end of this template strand at 3, 5 strand. In replication fork, RNA primer lies at the fork end of this template strand, 5, 3 strand. Formation of RNA primer constitutes the initiation of phase of DNA synthesis. How? Because without the presence of RNA, without the presence of RNA primer, DNA polymerase cannot add nucleotides. During base pairing, deoxyribonucleotide triphosphate come to lie opposite to the nitrogen bases of unwinded or exposed DNA template. Base pairing being ATGC, that is DTTP opposite adenine, DCTP opposite guanine, DATP opposite thymine, DGTP opposite cytosine. The two phosphates from the phosphorylated deoxyribonucleoside are removed and change them into monophosphate state by this enzyme, pyrophosphatase. Energy released during the conversion of deoxyribonucleoside triphosphate to be deoxyribonucleoside monophosphate is used for the establishment of hydrogen bond between free nucleosides. The process of copying genetic information from one strand of the DNA into the RNA is termed as transcription. If total DNA of organism is duplicated, it is called replication. Only a segment of DNA is copied in transcription. Only one of the strand is copied in transcription. Both strands are copied in replication. Copied strand and replaced with uracil instead of thymine in transcription. Why both strands are not copied during transcription? Mark the reasons as true or false. If both strands act as a template, they would code for RNA molecule with the same sequence, and in turn, if they code for protein, the sequence of amino acid protein would be the same. False. If both strands are copied, one segment of DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. True. The two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would promote the RNA from being translated into protein, and the exercise of transcription become futile one. False. If both strands act as a template, they would code in turn. If they code for protein, sequence of amino acid in the protein would be different. True. The two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein, and the exercise of transcription become fertile one. True. A transcription unit in DNA is defined primarily by the three regions of the DNA: promoter, structural gene and terminator. Enzyme helpful for the transcription: DNA dependent RNA polymerase. DNA dependent RNA polymerase. During transcription, the strand of DNA which has the polarity of 3,5 acts as template strand. During transcription, the strand of DNA which has the polarity of 5,3 acts as coding strand. DNA dependent RNA polymerase catalyze the polymerization in only one direction, that is 5,3 prime. 5,3 prime. DNA which has the sequence same as RNA except thymine at the place of uracil is called coding strand. Polarity of template strand 3,5. Polarity of template strand 3,5. Polarity of coding strand. Polarity of coding strand 5,3 strand. Polarity of mRNA. Polarity of mRNA 5,3. Complementarity does not mean identical. Answer the statements as yes or no. Yes. Promoter and terminator flank the dash in a transcription unit. Flank the structural gene in transcription unit. With reference to the polarity of coding strand, promoter is said to be located at and it's considered as phi end upstream. With reference to the polarity of coding strand, promoter is said to be located at and terminator is said to be located and it is considered as 3N downstream. Upstream or 5N, downstream or 3N, based on the polarity of which strand above the names are given. Coding strand. mRNA containing base sequences are complementary to this strand except thymine replacement as uracil, that is in template strand. mRNA containing base sequences are similar to this strand, coding strand. Template strand, write the mRNA sequence for this 5 UAC UACG. That is at 3N. Coding strand for this, write them mRNA sequence. UACG